Hi, my name is Jeon. I'm a postdoctoral researcher at Water Research at the Research Center, and I did my PhD in civil environmental and construction engineering at the University of Hawaii Manoa and graduated last fall. And I'm currently doing a lot of stuff, but I was trying to uh, present my PhD work. It was just enhancing wastewater surveillance for respiratory virus. Sorry for like talking about wastewater during lunchtime, but yeah, I hope you can enjoy. Okay, I was trying to explain like how COVID-19 pandemic affected my PhD years. I came here in 2020 August, which we're having a hard time, and there was a lockdown situation. And the first thing I came to the airport, I had to sign a bunch of forms that I have to be quarantined at a hotel in Waikiki. And if I violate this law, I have to pay either $5,000 or they're gonna send me to prison. So I have no choice to stick in a hotel for 2 weeks, but eventually I stayed there for like 6 months because I could not get any like accommodations during back then. But after that, I finally came out. Waikiki was like fully wiped out, like no people were there. Like, and I was like curious that well, how this virus, what single virus can change the whole world this much. And during back then, our lab had a NSF funding for COVID-19 wastewater surveillance project. So we were getting wastewater samples from like wastewater treatment plants, and we were like getting wastewater from like Manoa area to check the neighborhood situation. And I got like really interested in and participating in this project, and I eventually got talked with my PI that I could do a research with this for my dissertation. So if I talk a little bit about concept of wastewater surveillance as if you get COVID or like any kind of like virus, you'll get symptom or like you get infection and like there's like several days of infection period, and then you get a symptom onset. But before like symptom onset, you'll still get like your body will produce virus, and these virus could be shed through feces, mucus, like or any kind of body fluids, and that could be ended to sewer area. And then when we get wastewater from the sewer area and concentrate this virus, and then do a nucleic acid extraction, and we can detect how much of concentration of the SARS-CoV-2 is in the wastewater, or we can do a sequencing to know what kind of variant is in there. And then we can compare with the clinical case data or report to the public health. And the CDC know that the wastewater surveillance is important, so they conducted national wastewater surveillance system so that they could, and around more than 1,300 sampling sites are contributing to this to news, and it's covering about 44% of the total U.S. population, and they're not targeting only for COVID, they're targeting for other respiratory viruses, gastrointestinal viruses, or even fungal diseases like Candida auris. And if you can see from the left-hand side of the map, you can see that the sampling sites that contributing to the news have been significantly increased since 2020. And from state of Hawaii is currently participating in this too. And I did 6 months of internship at Hawaii Department of Health during my PhD year, and I conducted like multiple projects with them, and I developed like bioinformatic like tools for them so that they can easily report to the public. And but there are lots of research gaps for like wastewater surveillance back then. But during 2020, there are a lot of things we don't really know about SARS-CoV-2, and we don't really know about wastewater surveillance. And so I picked up 4 different research gaps. And first of all, I'll talk about the spurious correlation due to trends when you're correlating with clinical case data and the wastewater data. So if you are getting clinical case data and wastewater data, it's gonna be time series data, which means you're gonna have COVID-19 case data by time and SARS-CoV-2 RNA abundance by time. So if you want to compare these guys two, and you want to know if they're having a correlations with each other or which data comes faster or not, because because people can shed a virus before symptom onset. So if wastewater can catch the signal faster than the clinical case, it will be really important for us to take action faster. So, but if these two time series data have like trends like this, and you're not gonna have like it's gonna be hard to figure out which data is coming faster or not. So if you look at the lag 0 here, you're gonna see these both are having a trend. So if you do a correlation analysis, you're gonna have false positive correlation because of the trend. And if you go to lag 2, you can see they're not correlated each other, but because of the trend, it's gonna say, oh, it's positively correlated. And if you go to lag 4, you can see that the clinical cases is 4 days behind the wastewater data, and it has a correct positive correlation. But because of the trend, it still is still there, it's still affecting the correlation. So because of these trends, we call this false positive correlations as spurious correlations. And we need to remove the trends, we use a method called pre-whitening. This pre-whitening method is a detrending method. If you have a trend data and you use first differences between the time points, and then if you wrap it up, you can see that the trends were removed, but you can see the fluctuations between the time series time points are just remaining over there. So we use this to use it for our wastewater surveillance data. So for SARS-CoV-2 genes, we selected 3 genes, N1, N2 from the nucleocapsid gene, and E gene from the from the envelope gene. And then for the wastewater samples, we use liquid. We kind of separate into liquid and solid because we didn't know which sample would have a higher concentration. But and then we looked at the data that clinical cases were like dropping because this was after the lockdown. So the clinical cases were dropping, and all the wastewater data was dropping as well. So the both data were having trends. So if we did cross correlation analysis to see which data points are having which if clinical cases are having a lag, but because of the trends, it was showing a strong positive correlation between the clinical cases and the wastewater data among like all the lag. So when we do pre-whitening, we could see that the spurious correlations were all removed here. So, but we were not able to see the which lag were having the significance very well. So we use a normalization method. So this normalization method, we use fecal viral indicators. We use F-specific RNA call-phage that group 2 and group 3. They are usually found specifically in human feces. And pepper maltodvirus. This is a virus that infects a pepper. So when we consume pepper, it will shed this virus. So when we use this virus to normalize it, these viruses can be if there is a high concentration of these viruses, then it means there are a lot of populations contributing to the wastewater. So after normalization, we saw that from 2 wastewater treatment plants, clinical case data was 2 days behind the wastewater data. And then we try to check which normalization method is improving the cross correlation coefficient. And we saw that the F-specific RNA call-phage G2 and G3 were showing the best improvement of correlation, because I think we think that the G2 and G3 are from like human, they live in human intestine, but for PMMOV, this is depends on like pepper consumption, so it could be bias depending on the hyperconsumption of population. So next research gap, we discussed about qPCR inhibition when we were detecting for quantification. If you look at wastewater, there are like so many stuffs contributing to wastewater, and there are like pathogens, like organic particles, and a lot of wastewater contains humic acids. And this is pretty hard to remove during the nucleic acid extraction. And so if you're having wastewater samples that have SARS-CoV-2 virus and other substances, you will need to check if this is pure, or if this has like a like detection inhibitors like humic acids inside there. Because if you do RT qPCR for SARS-CoV-2 detection, this humic acid could like inhibit either the RT reaction or the qPCR reaction, so that it's when you check the quantification, it could be lower than you expected. So we spiked in humic acid, like different concentrations of humic acid for different concentration of SARS-CoV-2 RNA for RT inhibition. And we saw that like RT process was not really inhibited because due to humic acid. But when we look at the qPCR part, we saw that the qPCR was really inhibited, significantly inhibited, and if you compare between the qPCR and RT process, the CT delta CT, the CT differences between the control and the inhibited ones were significantly different. So that we know that the humic acid is affecting the qPCR more than the RT part. And then we try to see if this is because of the DNA polymerase, or is it because of the template inhibition. But these models were not able to explain how why is this happening. So we look at the amplification curve, and you can see that the fluorescence intensity of the amplification curve was dropping when the concentration of humic acid was increasing. And then we plotted using a Stern-Bolmer plot. This is a plot, this is the equation to see the fluorescent intensity inhibition. And if you can see the fluorescent intensity was increasing as the humic acid concentration is increasing, no matter how the cycles are, and whether the template concentrations are. And then, so we want to use a mathematical equation to correct this CT changes. So we use a mathematical correction to correct it. And after correction, you can see that the CT values are not changing even with the concentration of humic acid increases. And then we use it as a synthetic wastewater sample to test it out. And we were able to see that the red is the SARS-CoV-2 with the wastewater sludge cDNA. And then you can see that the fluorescent intensity was dropping after a spike it in. But after correction, we can see the CT values were corrected, as you can see. And we wanted to know what is the mechanism of fluorescent quenching by humic acid. That when you have a quencher and a dye emission, like have an spectral overlap, the dye emitted from the FAM dye that we use for qPCR can be quenched by the humic acid. And the FAM dye was around 520 nanometers for the emission. And then when we look at like other papers, that humic acid, when the concentration of humic acid increases from 4 to 10 milligrams per liter, you can see that its excitation and emission of the humic acid was increasing. And then there were similar cases like 2 antibiotic substances where I show a decrease of fluorescent intensity when humic acid concentration was increased. Next research gap I want to discuss was a rapid detection of important SARS-CoV-2 mutation. So if you during the COVID-19 pandemic, you heard a lot of variants were coming out, like Alpha variant and Beta, like Delta, and like the current Omicron. And these variants we call it variants of concern back then. These or depending on the mutations going on in the spike protein. This spike protein is the part of the SARS-CoV-2 that attached to the human receptor. And if it has a mutation going on, it will change the protein structure so that it can bind, it could mutate more easily to bind to the human receptor so that it can increase its infection. So it's important to track like mutation of the spike protein gene, the S gene, for future infectious variants. So we developed an amplicon sequencing method to track this receptor binding domain part of the S gene here. And we did a nested PCR so that we can amplify more because some of the SARS-CoV-2 SARS-CoV-2 concentration in the wastewater are low, so we wanted to amplify more. And this was around 532 base pairs, so it's a little bit long compared to qPCR. So the limit of detection was a little bit higher compared to the qPCR. So that's why we use a nested PCR. And then we designed an internal amplicon standard so that we can check if there's any like inhibition going on during the amplification. We did sampling from wastewater sampling site from back of the Manoa area. So this area was covering around 8,000 to 10,000 people living over here, and we sampled from April 2020 to December 2023, and then we trying to check the concentration of the SARS-CoV-2 first, because using RT-qPCR, and during the sampling period, there was a modification of nucleic acid extraction kit from Kaizen. They developed inhibitor removal technology, and then we used that to check for our samples. And before using IRT, we could see that the concentration of the SARS-CoV-2, which was higher, was only detected during this area, this period without IRT. But when we use IRT, we were detecting SARS-CoV-2 from every single samples. So which means this IRT was removing a lot of inhibitors from the nucleic acid. And then we use amplicon sequencing to check if the amplicon sequencing was improved using the RT tube. And then the Wilcoxon analysis were showing that it was significantly increased before using compared to before using IRT. And then we checked the important mutations going on in the receptor binding domain of the S gene. And then we could, there was an important amino acid mutations changing from early 2023 to over from late 2023. And this amino acid mutation was, a lot of studies have determined that this mutation has showed an increment of affinity with the AC2, the human receptor domain, and it has a resistance to the antibody, and it has a evasion to vaccine. So it was able to check the important mutations from the wastewater. And then we compare with the clinical case data and the news data. They were covering a lot of population, most likely the population of the Honolulu city and county of Honolulu. But our was covering only about 8,000 to 10,000 people. So the variant composition here is not really similar, but the variants were detecting similarly, and the XBB 1.24.1, this variant was only detected from Manoa. So we can see that if you sample from a smaller area, you cannot represent the entire population, but it can check the what variants of SARS-CoV-2 is circulating in the specific area. Next, next research gap that I discussed was a low target loads due to a low population. So what I detected before, when we targeted like an area with low populations, we will have low viral loads of compared to big cities or wastewater treatment plant that covers a lot of population. So and there current studies are focusing on a larger population with higher viral loads, but there are lack of studies of with a smaller population with low viral loads. So we want to like improve the sensitivity of the wastewater detection. So we use a tampon as adhesive material for absorbing organic particles or organic substances. So this is called a passive sampling, and a lot of people are trying to use passive sampling over like or composite sampling or graph sampling these days, because this is really easy to use. You can just put on into a wastewater, and you can take it out 24 hours later, and the tampon's gonna continuously collect the organic substances. And then we employed this into a direct sewage outlet that captures all the sewage discharging from the building. So so that we can collect as much target as possible. And we use nanoparticles that has a magnetic charge on it. So it targets the specific virus, specific viruses. And then so when we use magnets to just isolate all these viruses at the same time, so we can reduce the bacterial genes inside like a wastewater. And then we use a targeted enrichment library prep. So it enriches around like 40 different respiratory viruses, and it enriches around about 40 length of genome. So it was not only targeting like SARS-CoV-2, we can targeting like influenza, RSV, and other viruses. And then we target nursing home, because and a nursing home is a residential facility that provides long-term care for elderly and disabled individuals who need assistance with daily activities and medical care. And this project was done when I was doing internship at Hawaii Department of Health, and HDOH, they were trying to help the nursing homes, especially small nursing homes that needs like special cares. So they employed this wastewater surveillance to this nursing home, so that they can take care of the SARS-CoV-2 like infections going on in that facility. Because the when you look at the age distribution from the population, the age of over 65 is a little bit lower than compared to the younger, but you can see the fatal cases of COVID-19 is significantly higher compared to the younger cases. And this is a SARS-CoV-2 and COVID-19 death rates. As the age increases, you can see the death rates increases as well. And not only like certain countries, but from a lot of countries were showing the similar graph. So checking COVID-19 situation from nursing home is really important, because because of these reasons. And we were doing a 1 year of wastewater surveillance from the nursing home, and we targeted SARS-CoV-2, influenza A, influenza B, and RSV from the wastewater. There were several detection of SARS-CoV-2, but we could not. These were not led to clinical cases. But when there was a period there, for COVID-19 for SARS-CoV-2 detection in a row for a week. And during this period, the COVID-19 cases have increased from the nursing home. And first it was just two staffs were having a COVID-19 symptom. And after 3 weeks after the wastewater detection, the residents got COVID-19 as well. And then we use the sequencing, the targeted enrichment sequencing to check the full genome of the SARS-CoV-2. The first 2 weeks of the wastewater detection, where the concentration was pretty low, so the coverage of the SARS-CoV-2 compared to the 30 kb lengths of the genome was only around 0.2% or 21.4% was detected. But it was able to know if it's Omicron, but it was kind of hard to know which lineage was this. But after the third week, the concentration increases like significantly, and we were able to get almost the full genome of the SARS-CoV-2, and it was able to see the pangolage of LV.1.5. And if you can see from here that there was a SARS-CoV-2 detection at the end, but this didn't lead to COVID-19 cases, and the concentration was low, so we were not able to sequence the SARS-CoV-2. And we checked after 3 weeks later, we sent the personnel to the nursing home. We got nasal swabs from the residents who got COVID-19, and most of them were having a high concentration, and the pango lineage was LV.1.5. It was the same lineage that we detected from the wastewater. And we look at the phylogenetic tree, the wastewater and the wastewater SARS-CoV-2 and clinical SARS-CoV-2 were similar to each other, and with low mutation differences. And we look at the S gene mutation differences too from the clinical samples and the wastewater sample. Even with the low coverage rate, the mutational patterns were similar to each other. And so but we have a question to ask, that did the wastewater result alone lead to infection controls for the nursing home? Because we were keep telling nursing homes that, oh, we got SARS-CoV-2 detection from the wastewater, but for the first 3 weeks, there were even after the infected staffs were like isolated, and there were no clinical symptoms from the residents. And the home, even they tested the home testing kits, but they were like keep getting negative results. But SARS-CoV-2 was consistently getting from the wastewater. So we don't really know how much they control the infections in the nursing homes, but they were kind of like they don't know if like who is getting the COVID-19 because they don't have any symptoms. And of course, not all wastewater detection has led to actual infection. So like if you look at the previous data, there were like 7 detection of SARS-CoV-2 that didn't lead to infection cases. There was 1 RSV detected from the wastewater, but there were no clinical symptoms. And influenza A virus were detected from sequencing, actually from sequencing, but the concentration was low, and it didn't really lead into the infection. And we got wastewater directly from the building, so this virus it definitely coming from the building itself, but it did not lead to actual infection. So if we want to build their trust, we need to increase the accuracy of the wastewater surveillance, and then we're gonna need to be reliable for them to take actions like quickly as soon as they get the results. So main takeaways from my research is, first, Pre-binding has significantly removed the spurious cross correlations and detected early detection of SARS-CoV-2 from wastewater than clinical cases. And the second, fluorescence intensity inhibition was the major inhibition patterns by cubic acids during qPCR. And then the third, we designed a S-gene assay that can detect multiple mutations in the receptor binding domain of the S-gene, SARS-CoV-2. And lastly, we figured out that passive wastewater surveillance can be even be used in a small nursing home with low population. So that's all for today, and I would like to acknowledge my my lab mates and organizations and for helping this project. And if you have any questions, please feel free to ask.